In this video we will discuss about the different types of antibodies and their functions. Good morning. Welcome to YouTube channel, BioConcept, Learn with Concept. In previous video we discussed about structure of antibody, and based on heavy chains, antibody was classified into 5 classes: IgG for gamma chain, IgA for alpha chain, IgD for delta chain, IgM for mu chain, and IgE for epsilon chain. The immunoglobulin G. Immunoglobulin G is a 7S immunoglobulin with a molecular weight of 150 kilodalton 450 amino acid sequences. It has a half-life of 23 days, which is the longest among all the immunoglobulins. IgG is the most abundant class of immunoglobulins in the serum, comprising about 80% of the total serum immunoglobulin. There are 4 IgG subclasses: IgG1, IgG2, IgG3, and IgG4, so numbered according to their decreasing concentrations in serum. The immunoglobulin G has 1 VH domain and 3 CH domain, designated as CH1, CH2, and CH3. The hinge region is situated between CH1 and CH2. Their functions vary as follows: IgG1, IgG3, and IgG4 are special because these are the only immunoglobulins with the ability to cross the placental barrier. They play an important role in protecting the developing fetus against infections. IgG3, IgG1, and IgG2 play a role in activation of the complement proteins. IgG1 and IgG3 bind with high affinity to Fc receptors on phagocytic cells, and thus mediate opsonization. IgG4 has an intermediate affinity for Fc receptors, and IgG2 has an extremely low affinity for Fc receptors. The next antibody is immunoglobulin M. Immunoglobulin M constitutes about 5 to 8% of total serum immunoglobulins. It is distributed mainly intravascularly. It is a heavy molecule, 19S, with a molecular weight varying from 900 kilodalton to 1000 kilodalton. It has a half-life of 5 days. IgM is basically a pentamer, composed of 5 immunoglobulin M subunits and 1 molecule of J chain. Each monomeric IgM is composed of 2 light chains of kappa or lambda, and 2 heavy chains of mu. The heavy chains are larger than those of immunoglobulin G by about 20,000 dalton, because of an extra domain on the constant region SCH4. There is 2 subclasses of immunoglobulin M, IgM1 and IgM2, which differ in their mu chains. IgM1 consists of mu1, and IgM2 consists of mu2 chains. The immunoglobulin mu chain is a 72 kilodalton, 570 amino acid heavy polypeptide chain. This immunoglobulin has 1 variable region, designated as VH, and a 4 domain constant region, designated as CH1, CH2, CH3, and CH4. The mu chain does not have a hinge region. A tail piece is located at the carboxy terminal end of the chain. It comprises 18 amino acid residues. A cysteine residue at the penultimate position of a carboxy terminal region of the mu chain forms a disulfide bond and joins to the J chain. There are 5 N-linked oligosaccharides in the mu chain of humans. Monomeric IgM, with a molecular weight of 180,000 is expressed as membrane-bound antibody on B cells. The biological function of immunoglobulin M is following. Pentameric IgM, because of its high valency, is more efficient than other isotypes in binding antigens with many repeating epitopes, such as viral particles and red blood cells. It is more efficient than IgG in activating complement. IgM is the first immunoglobulin produced in a primary response to an antigen. The immunoglobulin provide protection against invasion of blood by microbial pathogens. Deficiency of IgM antibodies is associated with septicemia. IgM antibodies are short-lived and disappear early as compared to IgG. The presence of IgM antibody in serum, therefore, indicates recent infection. It is also the first immunoglobulin to be synthesized by a neonate in about 20 weeks of age. IgM is not transported across the placenta, hence, the presence of IgM in the fetus or newborn indicates intrauterine infection. The detection of IgM antibodies in serum is useful for the diagnosis of congenital infections, such as syphilis, rubella, toxoplasmosis, etc. The next antibody is immunoglobulin A. Immunoglobulin A is the second major serum immunoglobulin, comprising nearly 10 to 15% of serum immunoglobulin. It has a half-life of 6 to 8 days. Immunoglobulin A consists of alpha-heavy chain. The alpha chain is a 58 kilodalton and made of 470 amino acid residue-heavy polypeptide chain. The chain is divisible into 3 constant domains, designated CH1, CH2, and CH3, and 1 variable domain, designated VH. Hinge region is situated between CH1 and CH2 domains. An additional segment of 18 amino acid residues at the penultimate position of the chain contains a cysteine residue, where the J chain can be attached through a disulfide bond. IgA occurs in 2 forms, serum IgA and secretory IgA. Serum IgA is present in the serum and is a monomeric 7s molecule with a molecular weight of 60 kilodalton. It has a half-life of 6 to 8 days. It has 2 subclasses, IgA1 and IgA2, which are 2 alpha chain isotypes alpha-1 and alpha-2. The alpha-2 chain has 2 allotypes, A2-1 and A2-2, and does not have disulfide bonds linking heavy to light chains. Differences in the 2 alpha chains are found in 2 CH1 and 5 CH3 positions. Secretory IgA dimer or tetramer and consists of a J chain polypeptide and a polypeptide chain called secretory component. The secretory component is a polypeptide with a molecular weight of 70 kilodalton. It consists of 5 immunoglobulin-like domains that bind to the Fc region domains of the IgA dimer. This interaction is stabilized by a disulfide bond between the 5th domain of the secretory component and one of the chains of the dimeric IgA. Secretory IgA is the major immunoglobulin present in external secretions, such as breast milk, saliva, tears, and mucus of the bronchial, genitourinary, and digestive tracts. IgA activates the complement by alternative pathway. Immunoglobulin A has many biological functions. It protects the mucous membranes against microbial pathogens. Binding of secretory IgA to bacterial and viral surface antigens prevents attachment of the pathogens to the mucosal cells, thus inhibiting viral infection and bacterial colonization. Breast milk contains secretory IgA and many other molecules that protect the newborns against infection during the first month of life. Secretory IgA protects our body against bacteria, such as Salmonella spp, Vibrio cholerae, and Neisseria gonorrhoeae, and viruses, such as polio, influenza, and reovirus. The next antibody is immunoglobulin E. Immunoglobulin E constitutes less than 1% of the total immunoglobulin pool. It is present in serum in a very low concentration. It is mostly found extravascularly in lining of the respiratory and intestinal tracts. Immunoglobulin E is an 8s molecule with a molecular weight of 190 kilodalton and half-life of 2 to 3 days. Unlike other immunoglobulins that are heat-stable, IgE is a heat-labile protein and can easily inactivate at a temperature of 56 degrees Celsius in 1 hour. The immunoglobulin has 2 epsilon heavy chain and 2 kappa or 2 lambda light chains. The epsilon chain is a 72 kilodalton and 550 amino acid residues polypeptide chain. It consists of 1 variable region, designated as VH, and 4 constant domain designated as regions, CH1, CH2, CH3, CH4. It has no hinge region. In human it has 428 amino acid residues. The immunoglobulin E have many biological functions. Immunoglobulin E shows type I immediate hypersensitivity reactions. Immunoglobulin E is responsible for the symptoms of hay fever, asthma, and anaphylactic shock. Immunoglobulin E binds to Fc receptors on the membranes of blood basophils and tissue mast cells. Cross-linkage of receptor-bound immunoglobulin E molecules by antigen induces release their contents to the extracellular environment, which causes allergy. Immunoglobulin E helps in antiparasitic defense by mast cell degranulation. The next antibody is immunoglobulin D. Immunoglobulin D comprises less than 1% of serum immunoglobulins. It is a 7s monomer with a molecular weight of 180 kilodalton. The half-life of immunoglobulin D is only 2 to 3 days. Immunoglobulin D has the basic 4-chain monomeric structure with 2 delta heavy chains, with molecular weight of 63 kilodalton each, and either 2 kappa or 2 lambda light chains, with molecular weight of 22 kilodalton each. Immunoglobulin delta is a 500 amino acid residue-heavy polypeptide chain consisting of one variable region, designated as VH, and a 3-domain constant region, designated as CH1, CH2, and CH3. There is also a 58 amino acid residue hinge region in human delta chains. The function of immunoglobulin D is it's present on the surface of B lymphocytes, and both IgD and IgM serve as recognition receptors for antigens.